Welcome to this technical guide on how to perform a STEMpredict assay to determine cord blood, bankability and optimization of peripheral blood mobilization collection. After September 29, 2013, it will not be a requirement by NetCordFact to use the CFU assay as a functional test. In this technical guide, I shall be demonstrating how STEMpredict is used to determine if a cord blood unit can be permanently banked and how the collection of peripheral blood mobilized by granulocyte colony stimulating factor, GCSF, or other mobilizing agent can be optimized. My name's Ivan Rich and I'm the CEO of Hemogenics, the company that produces the STEMpredict assay. STEMpredict is the most rapid stem cell assay available. It is part of a family of assays specifically designed and developed by Hemogenics to measure cell therapy products. STEMpredict is one of several assays that belong to the HALO family of assays, and one of many that uses a standardized and validated ATP bioluminescence readout. The other assays that have been developed for this area are HALO, HemoFluor, and Hemolite 96-PCA-EQ, which are non-methylcellulose equivalents to the classic CFU assay. They incorporate the same growth factor cocktails as the methocult reagents. HALO 96 SPCQC and its mesenchymal stem cell equivalent, Luminesc 96 HUQC, is used to ensure stem cell quality during processing and other procedures such as cryopreservation. HALO 96 PMT are assays to measure time to engraftment and monitor global patient reconstitution. HALO 96 PQR and its equivalent Luminesc 96 PQR for mesenchymal stem cells are potency, quality and release assays. These two assays are the only commercially available potency assays on the market that comply with regulatory requirements. FDA master files exist for both HALO 96 SPCQC and HALO 96 PQR. All of our ATP bioluminescence assays use the same readout principle, and STEMpredict is no exception. The principle of measurement is shown here. All living cells produce chemical energy in the form of intracellular adenosine triphosphate, or ATP. When cells proliferate or are inhibited from proliferation by cytotoxic drugs, changes in the intracellular ATP concentration occur that correlate directly with cell proliferation, cytotoxicity, cell number, and even apoptosis. After incubation of the cells, the ATP is released by cell lysis, and this reacts with a luciferin-luciferase reagent to produce bioluminescence in the form of light. This is measured in a plate luminometer. But before any sample is measured, the assay is calibrated and standardized. This process is described in a separate technical guide video called Setting Up the ATP Standard Curve and Controls for all Hemogenics ATP bioluminescence assays. Here we're going to focus on performing a STEMpredict assay. STEMpredict is available as a 1, 2 or 4 plate kit. The assay is usually shipped frozen on dry ice. The box has been sealed. Upon arrival, please inspect the box to make sure that it is not damaged. Remove the seal and open the box. There should be an assay manual, MSDS sheet and content sheet included. Make sure that all of the components of the kit are included and store the kit at -20 degrees Celsius until use. Please read the assay manual thoroughly. The assay is actually easy to learn and fast to use. This technical guide should help you further, but you can always contact Hemogenics if you have any questions at all. Some of the contents of the STEMpredict kit are shown in this picture. The kit includes the STEMpredict master mix and a control master mix, medium, ATP standard, ATP controls, and an ATP enumeration reagent. The kit also includes sterile 96 well plates for cell culture and non-sterile 96 well plates for the ATP standard curve. Also included are sterile adhesive foils to keep unused wells sterile, and of course the assay manual. The principle of the STEMpredict assay is to compare the growth of a stem cell population stimulated with specific growth factor cocktails to that of a background control culture that contains the same cells as the sample but no cocktail. After setting up the culture, the cells are incubated for just 3 days and the ATP bioluminescence measured. Within the 3-day incubation period, the ATP ratio between the stimulated stem cells and the control cells should be 1.5 or greater to predict sufficient future viability and stem cell functionality to allow permanent storage. In this picture, samples S1 and S3 do not exhibit the required ratio and would not be eligible for permanent storage. If using mobilized peripheral blood, a ratio less than 1.5 would indicate that the stem cells have not been sufficiently mobilized. Let's now turn our attention to performing a STEMpredict assay. First of all, we have to consider the cell sample preparation. Performing any cell therapy assay using an ATP bioluminescence readout, it is important that the cells are prepared as a mononuclear cell fraction, or as a preparation containing less than 10% red blood cells. Mobilized peripheral blood are prepared by apheresis and do not usually require any further processing. Depending on the cord blood processing, the red blood cell concentration can vary from less than 10% to greater than 30%. Samples with high red blood cell content should be further reduced. Red blood cells interfere with the assay readout, causing false positive results. It's also advantageous to use a mononuclear cell count, an MNC count, rather than a total nucleated cell count or TNC count. The latter contains dead and contaminating cells that dilute the stem cells and lead to erroneous results. In this picture we have 2 cord blood samples, namely S1 and S2, on the right side of the test tube rack. The cell concentration has been adjusted to 500,000 cells per milliliter. For each sample there will be 2 assay tubes, one for the control master mix and one for the STEMpredict master mix. 0.9 milliliters of the STEMpredict master mix has already been added to tube SP1. The STEMpredict master mix has been added to both sample tubes now. The control master mix has been added to one of the tubes. All 4 tubes have been filled and the cells can now be added to the master mixes. The cells from sample S1 have been added to the first set of STEMpredict and control master mix tubes. The cells from the second sample are now being added to the second set of master mix tubes. Cap the tubes and mix the contents of the culture master mix tubes using a vortex mixer. The cell concentration in the master mix tubes has now been diluted tenfold to 50,000 cells per milliliter. Remove a sterile 96 well plate from the assay kit and unwrap it and place the lid on the side of the plate. Dispense 0.1 milliliters into each of 6 replicate wells across the plate from the tube containing the STEMpredict culture master mix for sample 1. 0.1 milliliters of the control culture master mix will then be dispensed into 6 replicate wells directly underneath. Dispensing 0.1 milliliters of the master mix into each well will produce the final cell concentration of 5,000 cells per well. The culture master mixes from all 4 tubes have now been dispensed into the 96 well plate. Replace the lid and transfer the plate to the incubator. The cells are cultured in a fully humidified incubator at 37 degrees Celsius in an atmosphere containing 5% carbon dioxide and 5% oxygen. Culturing hematopoietic cells under low oxygen tension is advantageous because it reduces the production of free oxygen radicals, thereby increasing cell growth and plating efficiency. Note the time at which the incubation is started. Incubate the cells for 3 days from this time point. Prior to removing the sample plate from the incubator, perform the ATP standard curve and control procedure to calibrate and standardize the assay. A detailed procedure for this is provided in the manual, and a technical guide video on setting up the ATP standard curve and controls is also available. The sample can then be processed and the ATP measured. This is also detailed in the kit manual, and a technical guide video is also available. Now let's take a look at some of the results from the STEMpredict assay. This graph shows results from a STEMpredict assay demonstrating the ATP concentration results in the upper graph and the corresponding ratio results in the lower graph. Note that the ATP concentrations for the controls do not increase, whereas the ATP concentrations from the stimulated stem cells increase with time in culture. The corresponding ratio values are shown in the lower graph and indicate that at 3 days of culture, the ratio of the stimulated stem cells to the controls has increased above the 1.5 level and increases even further on day 4 of culture. If the stimulated stem cells do not exhibit a sufficient increase in proliferation or remain at similar levels to the controls as determined from the ATP concentration, the stem cells will not be viable and able to sustain proliferation. Under these conditions, a cord blood unit should not be banked and stem cells will not have been mobilized. This concludes the technical guide for using STEMpredict. If you have any questions, please contact us at Hemogenics. We'll be pleased to help you. Thank you.